Hello, everyone, and welcome to today's webinar titled Monitoring Intact Light Chain of Therapeutic Monoclonal Antibodies in Human Serum by LCMS Mass Spectrometry for Clinical Research. This webinar is part of the 13th Annual Clinical Diagnostics and Research Virtual Event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during this presentation. You can find that live chat located on the left of your screen. Now, you can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. Now, you will notice that this speaker has provided a survey located at the right of the Ask a Question box. This survey will be available during both the live session and the on-demand period. If you have any trouble seeing or hearing this presentation, just click on that help desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located in the abstract tab from the menu at the left of your screen and follow the process to obtain those credits. I now present today's speaker, Dr. Yvonne Song, a Senior Application Scientist at Thermo Fisher Scientific. For a complete biography of our speakers, please visit the Presenter tab from the menu at the left of your screen. Dr. Song, welcome. You may now begin your presentation.
Good morning, everyone. I'm Yvonne Song, Senior Application Scientist for the Clinical Research and Toxicology Marketing Group at Thermo Fisher Scientific. Today, I'll share one of our applications titled as a monitoring intact light chain of therapeutic monoclonal antibodies in human serum by LCMS for clinical research. So here's my outline today. So I'll start with some background on the LCMS-based targeted protein analysis, monoclonal antibodies, MAPs, therapeutic drug monitoring, TDM, and some current techniques for TDM of MAPs. And I'll introduce our MAP workflow, intact light chain quantitation by LCMS, and I'll show some results and then finish with a summary. So start with the background. So what is mass spectrometry or MS? So mass spectrometry is an analytical technique that measures mass to charge ratio, MG ratios of analytes. There are mainly three components: ion source, mass analyzers, and the detectors. So first, samples are introduced by inlet source, for example, like direct infusion by syringe or liquid chromatography as a system. And those enter to the ion source here, where analytes are ionized by different ionization technique to create the charged particles. Then ionized analytes enter the mass analyzer. Under this mass analyzer, ions in gas phase separate according to their MG ratios. And by the detector, the ions are recorded further according to their MG ratios and the intensities. And mass analyzers and detectors operate under the high vacuum. So after data system, here is an example of the mass spectrum. So MG values on the x-axis and their intensities or relative abundance on the y-axis are recorded. And oftentimes, liquid chromatography or LC system is coupled to the mass spec. So LCMS analysis gives the three-dimensional information as shown in this graph, in this picture in the center. So you get the analytes first separate across the different retention time by the liquid chromatography, and for under each peak, their analytes further separated according to their MG ratios by the mass spec. And please keep in mind that mass spectrometry is different from the mass spectroscopy. And there are different types of mass spectrometers based on the various mass analyzers, and each mass analyzer have a different resolving power. So mainly here I introduced the two types called unit mass unit resolution mass spectrometers and high resolution mass spectrometers. So unit resolution mass spectrometers offer the mass accuracy generally between the zero point 0.1 to the 1 Dalton, which means you can separate analytes between these mass accuracies. For example, 1 Dalton mass accuracies. So if it's a less than 1 Dalton, unit resolution mass spectrometer doesn't have enough power to separate the two compounds. So examples of the unit resolution mass spectrometers include the triple quadrupole QQQMS or ion trap mass spectrometers. And you see in this nice animation how it shows actually how triple quadrupole mass spec detect the ions. For example, especially for the targeted quantitation. In this case, it's just only the filters and they detect the red ions in the red, and then operates at different voltage between the poles just to selectively quantify the red ions. On the other hand, the high resolution mass spectrometers offer the high mass accuracy, typically less than 5 ppm. ppm here is a parts per million. So example of the high resolution mass spectrometers include Orbitrap, time of flight, TOF, and a quadrupole TOF, QTOF mass spectrometers. And here showing the example of the Orbitrap mass spectrometer, how this mass spec is detect and record ions. So for example, in here, ions are the Orbitrap mass spec is operated by the oscillating ions through this cylindrical electrode. So Mg values of each analyte are recorded based on by the harmonizing oscillation and in combination with a Fourier transform. So high resolution accurate mass or HRAM mass is very useful and powerful to resolve the isobaric compounds. So here's an example of how the HRAM mass can resolve these two isobaric compounds with almost identical masses, just so different from the second decimal point. So for example, under the chromatogram, and their retention times are so close each other. So you need another secondary separation tool to accurately assign and separate each other. So using just the mass spec with the resolution of the 10K, which is not enough to resolve these isobaric compounds. So across the different retention time, you have only one peak, which actually two compounds merge each other. And then the later retention time, which only just a fluoxetine, but it doesn't have enough resolving power to accurately assign this compound. So you can see that relatively low accuracy, which is about like 95 ppm. However, when you use a high resolution mass spec, high resolution here is actually showing the resolution at 100K. So you have a nice separation and resolve these two compounds across the different retention time. Not just it offers the qualitative information, it offers also some relative quantitative information as well. So across the different retention time, you can see that relative abundance between the two compounds as well. So in this case, when you see it's kind of analytical challenges for the isobaric compounds separation, high resolution mass spec, HRAM mass spec is a definitely solution to resolve the analytical challenge qualitatively and then quantitatively. So there are different scan modes available by the HRMS, and I'm going to show just the three main types here. So first is a full MS, so which means that it scans all observed precursors at a certain retention time. You can extract the precursor ions of interest after the data acquisition, and it relies on actually high resolution of the photo selectivity. It's quite useful for the less complex background, and because it monitors all the observed precursors. So there is a no or minimum level of the method development before you run the full scan. And the second type is a TSIM, targeted selected ion monitoring. So as you can refer from the naming, it actually just target the certain precursor ions or analytes for during the analysis. So of course, if you want to target a certain analytes, you need MZ values, and you can also put the retention time just to selectively monitor the time window. It's powerful in the medium complexity metrics because you need some prior information before you run the mass spec. So you need some level of the method development is necessary before you run this TC mode. And the last type is TMS2 or parallel reaction monitoring, PRM. And with the TSIM, it's similar to the TSIM, but additionally it fragments your precursor ions. So you have, with the MZ values and the retention time, you also need a collision energy to fragment your precursor ions. So you can use those fragmentations, fragment ions for your targeted quantitation. This is more most selective method, even in the high complex matrices. And of course, you need more efforts before you run the TMS2 method. So there are mainly three approaches for the SCMS-based targeted protein quantitation, namely the bottom-up, middle-up, middle-down, or top-down. So top-down approach measure, analyze intake proteins. For example, here showing the IGF-1. And the second approach is a middle-up or middle-down approach, and it analyze the subunits or large peptides, usually higher than more than 30 amino acids. And for example, here showing the monoclonal antibodies going to actually subunit digestion or disulfide reduction and chemical reactions. So to create several subunits, so you can monitor different subunits by the middle up or middle down approaches. The last approach is a bottom up approach. So it's typically digest using enzymes, for example, trypsin, to the peptides. And for absolute quantitation, you spike stable isotope labeled peptides to quantify native proteins or peptides. I added one more approach here. This is a global profiling of your proteins. This is a relative quantification of many proteins. Here showing the plasma profiling. So you can quantify many proteins between the different sample types. As I briefly mentioned, here we monitor the monoclonal antibodies. So I'm going to give you more backgrounds on the monoclonal antibodies or MAPs. So these molecules are known as immunoglobulin. It has a very unique inherent specificity that recognizes and counteracts foreign substances, so-called antigen. In the early 1980s, it was actually first introduced as experimental therapy. And then that publication actually called monoclonal antibody as a magic bullet because of its specificity. It's shown in the picture on the right side, has a very unique structure, a large Y-shaped protein of the immune system, and has two main components called a light chain and heavy chain. And there are two types of light chain called a kappa or lambda, and a heavy chain, mainly the five subgroups and depends on the how many disulfide bond and how they structure each other. That's what the subgroups are typed. And therapeutic monoclonal antibody therapeutic maps are predominantly IgG1 kappa type with the size around like 150 kilodalton. So it's a pretty huge protein. And I'm going to also show that how the maps are named, because personally, I thought the map naming is quite tricky, but they're actually standardized nomenclature of the maps. So it starts with a prefix, some prefix, usually provide the manufacturer, followed by the what's the target or disease. So for example, Li or Lim for lymphocyte or immune, Tu or Tum for tumor. And they're also put what's the source of your monoclonal antibody. So there are actually 4 types of source. So here O for murine, and like Ki or C for chimeric, Zu for humanized, and U or U for human, so fully human. So for example, when you look at the adalimumab, this is adalimumab actually targets the immune disease. It's a fully human type monoclonal antibody. And the trend of the manufacturing maps is more toward to the actually fully human type because of the low potential for the immunogenicity. So immunogenicity is our natural immune response to the foreign substances in the body. So what it means actually, if you have your map drugs is a fully human, so less chance of your body react to the drugs. And shown in number here. This is actual percentage of the global market shares in the 2020. And here's some comparisons of between the small molecule drugs and the monoclonal antibodies. As you can see earlier, monoclonal antibodies have a very distinct characteristics. So again, the molecules is really huge, size of around the 150 kilodalton. And this is a protein, so it should be culture derived, and which means it require the multiple purification and quality control steps to ensure the consistency. And again, the specificity is very high, and also immunogenicity is low, but sometimes there's a immunogenicity happens as well. But also the administration is usually by the injection, so oral administration is not common for the monoclonal antibody drugs. And the dosing frequency is quite longer compared to the small molecules, so it can be every other week or 2 yearly. And here's actually the comparison between the aspirin, which is the common small drug, and to the actual size of the monoclonal antibody. And when you look at the market, it's a huge market because since 2010, that market has continuously grown, and with the expected market value by 2026 is about $209 billion. And when you look at the type of also I'm sharing the global market shares of the antibody drugs by disease, and the top share actually the autoimmune disease drugs occupying the 37%, followed by the solid tumor, other cancer type drugs, and also there's other disease type drugs which occupying the 23%, including the COVID vaccine. Indeed, the adalimumab or Humira in brand name is the top selling drug, except for the COVID vaccine currently. And I'm going to switch a gear a little bit. So because for the clinical use, therapeutic drug monitoring approach, or TDM, is very useful during the patient therapy for the clinical decision making. So TDM is the quantitative measurement of the concentration of the drugs in the blood, plasma, or serum. Currently, the measuring the concentration is the easiest way. It's the easiest way and a useful approach, reflecting the drugs outcomes. So by measuring the concentration using the TDM approach, clinicians can individualize dosing and optimize the patient responses to drug therapy. Because, for example, in this box, the picture, patients, this is actually kind of a pool of the patients with the same disease and the same drug treatment. However, not all of them show the same response. So most of them are going to show actually the predicted good response to the tested drug, but some people are going to show maybe poor or non-response, or sometimes fewer cases actually increase the toxicity risk. So clinicians have to decide either they have to change the dosing or frequency, or even change it to another drug, right? So by the TDM, measuring the concentration, if there's any toxicity or lack of efficacy observed, they have to adjust the dosing or dosing frequencies to get the desired effect during the drug therapy. So MET monitoring is very important, both in the biopharma and the clinical laboratories. So I'm just going to show how biopharma monitor the maps, and also the clinical laboratories measure the maps during their treatment. So in the left side, through the entire cycle, manufacturing cycle, or development cycle, so from the cell line development, upstream process, downstream process, until the formulation or fill finish, the quality attributes, for example, glycosylation or oxidation, is monitored to make sure the quality, efficacy, and the safety of the drugs. So after the drugs manufactured and administered to the patients, now clinical laboratories measure the concentration. So the samples are collected, for example, like blood collected, and then measure the concentration of the drug, and to make sure within the drug concentration within the therapy range at the steady state concentration. So with that, after the sample analysis, actually the doctors interpret the results, and with the medical judgment, they decided what's the next next dosing, because if the concentration does not reach, within the therapeutic range, right? So this throughout this cycle in the clinical laboratory, TDM measure ensure the optimized therapy for the patients. And here's some comparisons of the current techniques for TDM of the map. So since we focus on the SCMS approaches, so I'm actually highlighting some components compared to the other assays, especially the immunoassay. So by the SCMS, there are two sub approaches, actually tryptic peptides or light chain quantitation, but both approaches generate actually the detection level as sub microgram per milliliter range. And for the mass spec, actually the cost of instrument is relatively expensive compared to the other assays. And of course, and also they require the high expertise to operate the system compared to the other instrument. However, mass spec doesn't need the specific reagent to each or the target maps. So you can just apply generic reagents for the buffers or other consumables to measure the multiple maps in one assay. So the method is quite versatile. And also you don't need to upfront development, for example, like antibodies. So the upfront development cost is quite low compared to the immunoassays. And since the mass spec measures the molecule mass directly, so the specificity is really high. And again, as I mentioned in before, so you can apply the generic reagent. So multiplexing capability is quite high compared to the other assays. However, turnaround time is a little bit longer based on the typical SMS settings. But there is a new technique, newer systems like tandem LC or dual LC. So you can stagger the multiple runs, so you can save up to 50% of your turnaround time. And let's look at more closely on the mass spec-based assays. So here, protein and peptide quantitation approaches. So the first picture shows that the peptide quantitation approach. So typically you use a trypsin to digest maps to the peptides, and you target the signature peptide, which should be different from your endogenous IgG. And here usually used triple quad mass spec just for the targeting measurement of your peptides. And the lower panel shows the protein quantitation. So it generally reduces the disulfide bonds, and you can also add enzyme to digest to the subunits. And you target the intake light chain, or you can target the other subunits. But here shows the targeting the light chain here, and then use by the HRAM mass, you can quantify the intake light chain with the accurate masses. And of course, there are pros and cons for both approaches. So when you're using the peptide quantitation, there's a cost saving in the instrumentation, and then the method setup and the data processing is quite easy and manageable. And since you target the fragment of your targets, so you can also do the sequence verification. However, the peptide quantitation requires actually laborious sample preparation processes, oftentimes overnight incubation. And also you want to make sure to select the proper peptides based on the SCMS performances. And for example, also some maps have a very limited number of unique peptides. For example, like 100% like fully human type antibodies. So in this case, protein quantitation can be a solution because it targets a unique sequence and a mass of the map subunits. And by applying the HRAM mass, you can resolve the isotope of your proteins. And compared to peptides, protein quantitation is a relatively simplified sample preparation. So usually within an hour or a couple of hours, you can finish the sample preparation. However, the HRAM mass is relatively expensive compared to the triple quad. And also during the analysis, target maps often concealed by the endogenous IgG. And since it requires a high resolution mass spec data, so this generates a big data file size. So you have to make sure that you have a sufficient data storages. And in our study, we actually developed a method for the protein quantitation. the map intake light chain quantitation for the clinical research. So we have 4 aims. So during the development, we want to make sure to generate the fully resolved isotopic clusters by using the HRAM mass for accurate detection and quantitation. Second, we wanted to make sure to generate a reproducible chromatogram over the runs and the different column lots. Often we hear that for the protein analysis, there's some shift between the, especially the retention times. So we want to make sure to get the reproducible chromatograms. Third, we also make sure to generate a robust sample preparation method suitable for clinical use. And throughout the entire workflow, we also wanted to optimize, including the SCMS, considering the biological complexity of a human serum, while also we promote the fast turnaround time of the workflow. And this is the list of the maps evaluated in this study. So we target 6 maps, highlighted in blue here, from the adalimumab to the rituximab. We actually have a good combination between the oncology and the inflammation treatment drugs, and also we combine the legacy and the newly approved drugs. So for example, rituximab is the first map with oncology indication approved in the 1997. On the other hand, the carmalizumab, the third one here, is a relatively new drug approved in 2019 to treat the various cancers. And also we added 2 additional maps at the end, nivolumab and pembrolizumab, to use as internal standards. Because I'm going to show more details later, but just in short, because not all the maps have the heavy isotope version proteins, which should be used as internal standards. So we have to find the alternative approach. So we chose these 2 maps to use the internal standards for the different molecular weight of the intake light chain. And we apply the magnetic beads-based sample preparation because this Dynabeads or Pierce magnetic beads is a very fast and easy purification technique compared to the agar beads. So you just need a magnet without using the centrifuge or vacuum manifold. So you just can use a magnet to separate beads with the buffer or sample. And this kit actually has the include the buffers, so you don't need to prepare the buffer like washing buffer, elution buffers. So, and with the different ligands available in this kit as well. And also Kingfisher system is an automated purification system with the different portfolio. So for example, DuoPrime is a simplified platform. And here the Presto is actually can be added to the Hamilton system as an accessory. So you can just easily apply the magnetic beads sample preparation. And also the automated scripts are available online, so you don't need to have to spend a lot of time to write the scripts. So I'm going to show some data from our optimization process. So here first, we show that what purification technique we choose and also how we optimize the sample preparation. So we tested different purification techniques from melon gel, Dynabeads with the different ligands like protein A, G, and L. And also we test the agar-based beads. Here is a kappa XP. So protein A and the G, you can see at the bottom of the picture, protein A and G bind to the Fc containing IgG, which is a heavy portion of the heavy chain. And the protein L and kappa XP bind to the kappa light chain of the IgG. So after purification, we add an IDS enzyme to digest, to cleave the hinge regions, the heavy chain, and further through the disulfide bond reduction, we generate the 3 subunits, including a light chain. All the purification techniques are capable of the high throughput and automation. It's just a different platform like magnet or centrifuge. And this is just one data between just the protein G and protein L comparison. And then I want to mention that the same samples were used and both purification and SCMS analysis were performed on the same day. As shown in this graph on the right side, so orange bar indicate the peak areas using the protein L magnetic beads. The blue indicate the protein G data. So as shown in this graph, the purification yield is higher for the protein L, except for the golimumab. And this actually confirmed by the repeat. So we finally chose the protein L of our purification technique. And I like briefly mention we have actually the alternative internal standards approach because heavy labeled version of the maps are not always available. So we investigate the other approaches, including other maps to use as an internal standards. And we chose that nivolumab and the pembrolizumab to use as for the different mass spaces of the target map. So 1 microgram, and you can see that how we assign these two internal standards to the target maps here in this table. So 1 microgram of these internal standards were spiked to the different concentration on the map, as indicated in this table, to generate the calibration curve for the analytical performance evaluation. And we also optimize acid conditions. So for example, here we added 0.02% TFA, trifluoroacetic acid, and IPA, isopropanol, to help protein elution and carryover removal. So this is the final composition of our mobile phase with a column, which is a map pack reverse phase column. And we ordered the 3 column lots for the quick column reproducibility evaluation. And it's shown in this bottom side of the graph. It's a gradient we use for this workflow, and as a result, um, there's a nice separation between O6 maps across the different acid gradients shown in this chromatogram. And for HRAM mass spec, we used the Orbitrap Exploris series, and there are actually 3 Exploris series available. And then this number 120, 240, and the 480. This number indicates the max resolution that can reach to. So here 240, for example, 240. This instrument can reach the max resolution of the 240K. And we also apply the intact protein options for the optimum protein analysis. So why you apply the intact protein application? Because there are different pressure settings, like trapping gas pressure settings, optimal for the different size of the molecule. So here we apply the low pressure mode, showing in the middle. So this is the low pressure. This low pressure is optimum for the subunit and the top or middle down analysis. So it is shown in this spectrum inside. The blue with the low pressure mode result in a significant improving the signal comparing to standard mode, which is almost 4 times higher. So we're using by using the Orbitrap Exploris 240, we have a nice fully resolved isotopic clusters of the different subunits. So the first chromatogram show that the separation between the 3 subunits, and for each peak there is a resolved isotopic clusters in the middle. And again, the data is acquired with the low pressure mode for the intact protein application. We also test the different resolution in the mass spectrometers, and as a result, 120K is the optimum resolution to accurately assign and quantify the size of the subunits. So if it's lower than the 120K, you can see from, for example, the 60K, so you cannot actually accurately resolve these different isotopes. And for the data processing, we use a trace finder, one single software from the data acquisition to the reporting. So with the trace finder, you can also put the different operator levels. So each user can acquire their own sets of the data or batches. So you can generate the methods and also acquire the data on the batches, and then also do the data processing to the reporting using this one single software. And further, we optimize the mass spec method because I don't want to target the whole entire series of the isotopic clusters. So I wanted to really focus on the certain ions to to gain the signal intensities and the accurate quantitation. So after confirmation of your molecular mass using the biopharma finder, I further adjust the scan range to the 1,000, just scanning the 1,000 to the 1,400, just to target the most intense charge states. So for example, on the right side, I target the charge state from 20 to 23. And in each charge state, I also picked 3 most intense ions from the 4 different charge states. So a total of 12 mg values were put into the trace finder for the peak extraction and further quantitation. So after this optimization process, here is the final workflow. So just a 10 microliter of samples were taken and subjected to the protein L magnetic beads purification. And we also implement the on beads sample treatment. So you don't have to transfer between the different tubes or plates. So you just have to apply the buffer and the enzyme reagents. So after the IDS digestion and also the disulfide bond reduction and generate the 3 subunits, right? And then these elutes are subjected to the SCMS analysis. So again, this workflow just need a low volume of sample, which is a 10 microliter per analysis. And with the sample treatment on the beads, this method just offers a minimized sample loss. And also using the trace finder software from the data acquisition to the data processing and reporting. So it's a seamless data workflow. So after generating, by generating the calibration curve, this is a result of the analytical performance evaluation. And you can see that all the 6 maps showing the great linearity with the R-square values higher than the 0.99. And the LOQs were determined to be between the 1 to 5 microgram per milliliter in a human serum. And I added the 2 additional graphs on the right side. These are actually the peak areas of our internal standards, which is showing that the RSD values is less than 12%, and it supports actually a very reproducible workflow from the sample preparation to the SCMS analysis. And also one of our aims to generate the reproducible chromatogram, right? So we graph the observed retention times of the 2 internal standards over the entire calibration curves on the left side. So you can see that the retention time variation is only the plus minus 0.05 minutes across the entire calibration analysis. And on the right side, I put the example of the overlaid chromatograms of the pembrolizumab from rituximab calibration curve sequences, and you can see that these peaks, all the peaks actually nicely overlapped between the different injections. And as I mentioned, we also performed the quick evaluation on the column reproducibility. We originally ordered 3 column lots, but unfortunately, there's 1 column lot backlot, so we have to just include 2 columns from 2 different lots in this study. So again, please remember that this is not a full validation report, but we wanted to show that this MapPack column can generate the coherent data for your protein analysis. So overall, peak areas were not significantly different between the between the columns. Especially the reproducible retention time was observed within the same lots, showing the 0.05 minute difference. Between the different lots, there's a slight retention time shift, showing the 0.2 minute difference. However, this can be observed also when you prepare the new buffer. So this is considered an acceptable shift. And when you see on the bottom side, there is actually summary of our column reproducibility evaluation, showing what's the average retention time, peak areas. Also, we put the percent peak area difference between the max and mean values. So in summary, based on our aims, we successfully developed the middle-level approach for the tech light chain quantification of therapeutic maps. And with the high resolution mass spec, we applied the resolution higher than the 120K with the low pressure mode of the intact protein application in the Orbitrary Exploris 240 mass spec. Also, we achieved the reproducible chromatogram, showing that the retention time, reproducible retention time in the peak areas as well. And from after the successful optimization of the workflow, we achieved just a low volume of sampling, which requires just a 10 microliter sample per analysis with a 1.5 hour sample preparation and 12-minute LCMS analysis. So as a result, LOQs were determined between the 1 to 5 microgram per milliliter. And if you're more interested in our the details, you can scan this QR code right in the middle and to download this technical note. Finally, I want to thank our team members who help on this technical applications, and also other clinical research, toxicology, marketing team members as well. This is definitely the teamwork, and I really thank our team for the support.